Hey, welcome to our cells and grossology class. This was a former workshop. It was all about cells. We decided to juice it up a bit, and we added some stuff from a summer camp program that we do called Grossology, the science of the human body. So it's kind of related to cells. We're going to try to relate it to cells, and we're going to do some. The kids are going to get a kick out of this. So setup for the class is one of the gross things we're going to do is talking about gas. And so we want to take one of these hand pumps and we're going to pump up a whoopee cushion. You want to do this before class or without the class seeing it if they're in there. And you want to just pump it up. Not to be all the way, that's probably good right there. And then we want to do is we want to set it somewhere like on a chair or somewhere a kid can sit without the kids in the class seeing it. So maybe if you have a chair, you can turn the chair around and then put it so the kids don't see it, and we'll lead a child up and they'll sit on it later. So we're just gonna put this over here for right now. Put this over here. Other setup for the class is we want to make some Jello before you get to class, like the night before, because the take-home is going to be some Jello. That's cytoplasm. It's going to be an edible cell. So this is the container you want to make the Jello. You want to figure out how much jelly you need.
So this is take home for Grossology. You can see I got the cytoplasm right here. You have to do this night before. This takes a while. So what you do is you follow the instructions on the box. It says use the entire packet. I poured the entire packet in here. You boil water. You put a cup of water in here. You stir for at least 2 minutes. That is what I have done. Next step is to put some cold water in there. Stir that up, and then put it in the refrigerator and wait. And it will set up overnight. All right. So we are expecting to get 2 cups worth of material out of here. You are going to get a 1/4 cup measure to spoon out to the kids. You're going to want to be very careful that you don't give too much or make sure you make enough Jello, because you should get about, per package, about, what, 8 kids worth. So you can make sure you make enough for whatever number of kids you have the next day.
So 8 take-homes per box. What size box is that?
That's the small little one like that. Just like that. I got one over here. Yeah, there we go.
We got a 16-ounce box?
3-ounce
box. So you would need, for a class of 20 to 22 kids, you would need 3 to 4 boxes.
3 to 4 boxes. It says in the cheat sheet, as of right now, we'll try to change that. It says 2 to 3 boxes, but just do the math.
At least 3.
Yeah.
For a full class.
Yeah.
Smaller class, 1 to 2 boxes. Try to use the complete box and not store a partial box in the kit, because they could get gross. Is that your phone, Tom?
That would be my wife calling, because of course it is. Enjoy your Jello.
Okay, so that's pretty much set up for the class. Let's get into it. Introduction to cell. We talk about cells. There's something all living things have in common. Of course, we go through the rules before that. We give some cool facts about cells, how many there are, how thin they are, stuff like that.
Hi guys. Tom here to talk about cells and grossology, the acting in a cell activity. All right, first you're going to have to take the stuff out of your bag. You're going to get 2 boss laminates. Inside this mitochondria bag, there's 2 little light bulb necklaces. We'll take out in a second. These are the pipe cleaners, which we'll use later on. The lysosomes. It's got 2 buckets and 2 little trash can looking things, and one of them has a bunch of paper in there. Just a top. And these hats are very flimsy. We might be doubling these up because they're very flimsy. But these are our ribosome hats. Let's see if I can get this apart. Like I said, very flimsy. So, 2 of these. What we will be doing, and it depends on the size of your class, what we will be doing is, if you've got 11 or fewer kids, you're going to take one of each of these things. If you have more than that, you can double them up. All right? So, say we're doubling them up, right? First, you're going to get your kids to be your nucleus. These are going to be our boss. They're going to wear our boss laminates, okay? And the necklaces around them. And they're going to stand in the middle of the room or the middle of your large area that you've set up, okay? They're going to be the ones that are going to telling all the other kids what to do. They're going to hate that, right? Then you're going to have your mitochondria, and they're going to wear these little necklaces. They click on and off just from the top, and then they sort of flash different colors. They are the powerhouses of the cell. They provide energy, okay? Now, next we have our ribosomes. Our ribosomes they make amino acid chains. They take amino acid, put them in chains. That's part of what they do. So what you'll do is you'll give them a couple of these pipe cleaners, okay? Get a couple of the pipe cleaners and you'll have them start making chains, okay? And this is, once we start the activity, they can make a few chains, so the chains will look like this. All they gotta do is put them together, and then they can get another one, blah, 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 all right? So that's what their job is, but they're only gonna have a couple of pipe cleaners and they're gonna quickly run out once you start. Your lysosomes are the digestive part of the cell. They're the ones that take the food that comes in and they digest it and they turn it into energy for the cell. These are going to be the things that we're going to represent. These little paper balls are going to represent the food that comes in. So we've got 2 ribosomes, 2 lysosomes, 2 mitochondria, and 2 people wearing boss laminates, okay? And of course, there's only 1 nucleus in the cell, but it doesn't matter. 2 of them will be wearing that. Now, all the rest of the kids will form the cell wall. If they're not touching, that's even better, okay? So the cell wall will be all the way around them in the circle, and you can show how it's semi-permeable by either going underneath their arms or kind of like pushing your way through or whatever. So you talk about semi-permeable. So this is the food, and what you can do is you can actually pass out these little pieces of paper to the different kids, okay, on that, or the cell wall, and then on your command they can throw one in there. When they throw one in there, the lysosomes will scoop them up at the direction of the nucleus. The nucleus will have to say, hey, go pick that up. Hey, there's one right there, go get it. They'll pick it up. Once they pick it up, they can then give this over to the ribosomes, okay? And the ribosomes can then make a chain out of it after the boss laminate says, hey, the nucleus says, add that to the chain, add that to the chain, okay? Once they kind of get the hang of that, if they do a few of them, don't do them all, if they do a few of them, then what you can do is say, oh, and go over to like one of your ribosomes, your lysosomes, say, hey, you're getting tired. So they have to like fall asleep, tend to be asleep, and then the nucleus people will have to tell the mitochondria to go over there and give them some energy, and they do that by tapping them on the shoulder, and then they can get back up. Once they kind of get the idea of all this, then you can do it a little bit faster, and so they can act out the different parts of the cell. We're using these different parts, and there are of course a lot more. In fact, there's more information inside of your manual, and inside I think even on the cheat sheet it has more information of the different parts of the cell. These are the ones we're focusing on because at the very end when we do the take-home activity, that take-home activity has these parts, so we want to make sure that they understand what these parts do. The rest of it, if you have time, the Golgi bodies and all the rest of the stuff, you can go through that. And that is acting out a cell.
Okay, next we're going to talk about cells again and the process of osmosis. And osmosis means that cells have what's called semi-permeable membranes, which some things can get in, some things can get out, but a lot of things don't get in. I relate it to like a classroom wall, where if someone has a smell in a classroom wall, that's a permeable membrane, and we can't see the other wall, we can't hear anything, or we can't smell anything from there. But a semi-permeable membrane would be something like a balloon. So what we do is we take a balloon, we put a little vanilla flavoring into it with a pipette. Just bring a volunteer up, of course, to do this. And you can do that. The other way to do it is just drop it in, but the best way to do it is have them pipette a few drops of vanilla in here. Just put it in there. What you want to do is take the balloon, what you can do is have your volunteer kind of shake it around a little bit, and have your volunteer then smell it. So they can shake it, and you can smell the vanilla in there. And so you can, the volunteer can then go around to each group, maybe have them smell the vanilla. So the fact is, the vanilla still can't get out, but they can smell it. So that's semi-permeable, and it's kind of like cells, so it lets the smell out, but the liquid itself cannot get out. So that can be like a cell, we relate that to a cell membrane, something that's semi-permeable. Next, we also have a microscope you can set out. We are going to hopefully get some slides for this, so maybe during a lull part of the program, the kids can come up maybe and take a look at a real cell in a slide there. Next, we're going to move on to the gross part of this workshop, and the first thing we're going to talk about is We can talk about related to cells. We can say, well, the highest concentration of nerve cells in our body are in our brain. Okay, huge concentration of nerve cells, and there's a model of our brain, so you can show how the brain. That's a real size human model, good scale. You can take it apart and show the different hemispheres of the brain. But we're going to do something real fun here. We're going to make brains. You guys want to make brains? The kids are going to go nuts.
We're going to make this brain. We're going to make it better than this brain. We're going to make a brain like this, but better. All right, first we're going to start with sand. Okay, from your cheat sheet, get your mixing bowl. You got your sand. I like to do this step first because I scoop the sand out, and that way I don't get potato and everything. 1 cup of sand. Next, we need 1 3/4 cup of potato flakes. Now, this is a full cup. This line right here is a 1/2 cup. In between, someplace is 1/3 of a cup, so 3/4 of a cup. So eyeball it. We're not equal, so it doesn't matter. 1 cup. And whatever. Close enough. Now, we need some spice drops. And at this point, we're going to have to time lapse because I need to cut these spice drops. You should do this beforehand. All right, this is not quite a 1/2 cup, but you get the idea. So 1/2 cup.
Maybe you don't need a 1/2 cup.
Maybe you don't need a 1/2 cup, Sarah says. Whatever. Now, I like to have an official stirrer person to do this for you, so you maybe get somebody and put one of their gloves on, make sure everything's nice and mixed up. Now, you can either put the food coloring in the pitcher, or you can put it into this thing. I don't really care. You choose. I'm going to put a couple drops here. Okay. And I need a 1.5 cups of hot but not boiling. You want to start this before class starts.
Tom found putting the food coloring in there beforehand will help.
We might change this to red food coloring, we'll see. But definitely put your food coloring into the water before you put it into the brain mold, because it'll just get absorbed and it'll be all blotchy. Oh, exactly a 1.5 cups, look at that. You can see it's still steaming, that's fine, you know, doesn't really matter. Just, you don't want to have it so hot that you can't touch. Now, if you have a volunteer come up and do this, make sure that they're not that they're wearing a glove and that it's not too hot. Test it yourself first. This will not fill up the entire brain mold. It is not imperative that you fill up the entire brain mold. What you're trying to do is just get enough so you get something that looks brain-like. So, this is still a little bit blotchy. I'm going to stir it a little bit more just to get the yellow all the way through. Which, you know, whatever. So now you've got it all through. Now pour it into your brain mold. Woo. Nice. All right. Now at this point you're going to go, it's not enough, it's not enough. Don't worry, it doesn't matter. Kind of get it packed around there as best you can. I like to push it up the sides just a little bit, try to get as much as I can, looking brain and brain-like. Particularly worried about the stem there. Don't want that reptilian brain not to be represented. Science. All right. This point, I'm gonna make a loud noise. You guys ready?
Yeah.
We're turning out the brain. Ready? 3, 2, 1. Booyah! Booyah. Looks kind of gross with weird stuff in it. You'll have more spice drops than me. But you can see this is when you put the yellow food coloring, the red food coloring, whatever color we end up having. I think green would be all right too, because that'd be awesome. But this is what it looks like when you put it in with the water. This is what it looks like when you don't put it in with the water, because you see how blotchy it gets. Which
also looks cool.
Okay too. So up to you.
This is a diseased brain right here.
This one right here is your brain on drugs, and this is your normal brain. All right? Don't tell kids that. That will probably get you in trouble. Hi everybody, it's Tom. I'm going to be dealing with the Grossology kit. Specifically, the science of body gas. We are no longer saying farts because parents. Now, the science of body gas, we have a whoopee cushion. Whoopee cushions are very easy to destroy, so we're going to take a few seconds just to talk about the proper inflation level. Got your balloon pump, got your whoopee cushion. Stick this in here, make sure it creates some sort of seal, doesn't really matter. If it makes a noise, you could get it, whatever, kids are going to love that anyway. You fill it up. This, maybe one more squeeze, is about the proper level of inflation. You can see there's a little bit of looseness to it. It's not over-inflated. Can maybe give it 1 or 2 more pumps and it would still be within the range of what we consider acceptable. That's getting right about to the point where it's getting too much. Once you start to see the buckling on the sides, that means you're over-inflating. Now the kids are going to do this. Their kids are going to do this, and they're going to try to start stomping on these things to see how loud they can get it. All right. This will destroy them. This will bust the seams, and we don't want that to happen. All right. So if you see some kids doing that, just push some air out until it gets to about the proper level. It should be about like that. All right. At this point, you can place it on your chair. I have a chair here. I will put it there. And when you sit on it, it makes plenty loud enough of sound. Ooh. As you can clearly see. And that is the science of body gas.
Okay, so here's our fart bag. And on the back, the directions that say to squeeze it and then stand back. So we're going to feel around. There's a little pouch in there of chemical I've got to pop and throw. Oh, Jackie, watch out. Oh, God.
That sounds more like an explosion.
Oh, it was a fart explosion. So it explodes chemicals. So this is why we want to do it outside. And it smells terrible. Can you smell that on video? Oh, it's bad.
The next activity is called super spit activity, and it's an activity that kind of works with something that's gross. It's about spit. And so what we're going to do is we're going to use these little whale crackers, 1 bag per class. There should be plenty of crackers in there. You want 2 crackers per student. You're going to pass out the first cracker. And what we want the kids to do is we want them to blot their tongue and get it nice and dry. So they're going to stick out their tongue everywhere, and they're going to make sure it's dry. And then they're going to take a bite of one of these cheddar crackers and just taste it. And then the next thing they're going to do, we're going to come along with a pitcher of water. We're going to pour a little bit of water into a Dixie cup form. They're going to sip that water and get some spit going, and then they're going to eat their other cracker. And what they should find is when their mouth is wet and has a lot of saliva going, that they're going to taste much better than when they did without the spit and it was real dry. Saliva helps us taste things. It's also very important with other things. It breaks apart our food. It helps with digestion and stuff like that. So we explain that as we're talking about spit. So it's gross, but it does help us, just like the methane that we produce in farts. It's gross as well, but it does help us. It's decomposes food and everything. The take home is going to be extra gross. We're going to get back to cells now and talk about what we're going to do is we're getting now. You've got this Jello that's pre-made in there, and you've got a scoop to scoop it out. I think this is the scoop right here. You're going to use that. So 1 scoop per child, or about even half a scoop in there. The children, the best way to do this is kind of organize a little assembly line. The kids are going to get each a bag, and this bag is the cell membrane that holds the cell together, just like we studied in the cell parts. They're going to hold open their bag. They're going to walk up. You're going to give them a little scoop of Jello. The green Jello works great. That's their cytoplasm. Then they'll come back around again for an Oreo cookie, and that is our nucleus. It's got the layers on there. They're going to drop 1 Oreo cookie in there, and then they're going to come by again for a mitochondria, and mitochondria are spice drops or jelly beans we'll have in here. And then they're going to come back around again for lysosomes, which are M&Ms. They're going to come back around again. And now the M&Ms and the spice drops, you can put maybe 2 in there, 2 or 3 max, but just a couple. You don't want to dump a whole bunch in there. Ribosomes are either going to be red hots, which you're in here now, or nerds, something that's small and round. And again, that'll be our ribosome. So they're going to get their whole cell. They're going to Ziploc it to make sure it's safe. And we actually have I believe we have a sticker to put on there, a label.
The take home talks about it.
We're going to get labels in there that will put on what each piece is, so that when they show their parents, they'll see each piece of that nucleus. What kids like to do sometimes is mash it all up, and they say once they show their parents, they got to show their parents first before they can eat it. We don't want them eating this in class; they're just going to make a huge mess. Also, we do have candy and food items in this kit, so we want to be careful with this kit. Do not leave this kit in your car at all. We say that for all our kits, because what's going to happen is this candy is going to melt and get all messy. You don't want that to happen, especially in this heat. So definitely take this kit inside with you when you're not using it. Keep it at home. That's cells. I don't think we left anything out.